Okay, so we have thermoreceptors. They're phasic. So these are free nerve endings that are found in the dermis, in the muscle, liver, hypothalamus. They rapidly adapt. So there are warm receptors that respond to temperatures between 77 and 113 degrees Fahrenheit. Um, they are not as abundant as the cold receptors. So warm receptors, that information is carried on C fibers, which are the slow ones, remember. We also have cold receptors that respond to temperatures between 50 and 68 degrees Fahrenheit. This information is carried on A delta fibers, which are faster. They respond to stimuli within a temperature range that does not damage the tissue, right? Because if the temperature ranges are outside of these ranges or in a range that's potentially threatening, the body will recognize them as pain, not just as temperature. So they're very active when the temperature is changing, but they quickly adapt to a stable temperature. So once the temperature is stable, and you're in that stable temperature environment, they're rapidly adapting to that. So you seldom notice a room temperature unless it changes suddenly. Now, if we're sitting in the classroom and all of a sudden I crank up the thermometer and I make it super hot, then you will be aware of that. But then once it becomes kind of a steady state, steady temperature, you will adapt to that. Now, there are, of course, many more cold receptors than warm. And again, the temperature sensations are carried along the same pathway as pain. They're just sent to different areas. So the chemoreceptors, they detect small changes in concentration of specific chemicals and compounds. Generally, they respond to water and lipid-soluble substances that are dissolved in bodily fluids or body fluids. Things like interstitial fluid, blood plasma, cerebrospinal fluid. These chemoreceptors are looking at things like pH and CO2 level, oxygen level. They adapt over time, and that information is sent to the brainstem. Right, so then the brainstem can do something, hopefully can do something about it to regulate and bring you back to hemostasis. Mechanoreceptors are sensitive to stimuli that distort plasma membranes. So they contain mechanically gated ion channels. The gates open and close in response to things like stretching or compression or twisting. There are three classes of mechanoreceptors: tactile receptors, baroreceptors, and proprioceptors. So the tactile receptors provide sensations about closely related touch, pressure, and vibration. So fine touch and pressure receptors provide detailed information about the source of the stimulation. Things like the location, the shape, the size, the texture, movement. And they're extremely sensitive. So they have those really narrow receptor fields. Because it's fine touch, you're looking for really specific information. Now, crude touch, of course, you're going to assume those are larger receptor fields. So crude touch and crude pressure receptors provide poor localization. There's very little additional information about the stimulus. It's kind of a vague report, because those receptor fields are really, really large. So you can't localize exactly where you're feeling it and what. So looking here, these are some of the different types of endings that read superficial fine touch. So again, if it's fine touch, where it's smaller receptor fields, because it's really sensitive, because it's fine touch. It's not crude touch. It's not kind of vague. It's very specific. This information is carried on the A beta fiber, so they're fast. So here's an example of 4 of them. So on the top left, it's free nerve endings. These are scattered through the epidermis. They're sensitive to touch and pressure, and they're tonic. So these free nerve endings are located between epidermal cells. They have small receptor fields, as you can see in the picture. They are very similar to the free nerve endings that provide pain and temperature sensations. The interesting piece, our free nerve endings are the only sensory receptors on the cornea of the eye, which is interesting. So all these different tactile receptors range in complexity from these kind of most basic free nerve endings up here on the top left, all the way down to these more specialized sensory complexes as we move along. So then on the top right, we have hair follicles, so hair follicle nerve endings. So on all hairs, we have this root hair plexus down at the bottom. It monitors distortion and movement across the surface. When the hair moves, the movement of the follicle it distorts the follicle and the sensory dendrites and produces an action potential. So the hair follicle nerve ending is an example of one of the free nerve endings. It wraps around the base of the follicle. It's phasic and rapidly adapts. Versus the free nerve endings on the left are tonic. So we also have on the lower left these Merkel discs. So the Merkel discs are found deep in the epidermis. Sometimes they're either called Merkel discs or tactile discs. I've seen it written both ways, so just so you know, they're the same. So you could write there in your notes, tactile discs right next to Merkel disc, just so they're the same. So they have small receptor fields that make them very sensitive. And so this Merkel disc, or the tactile disc, comes in, it's like a big flattened dendrite that you can see there as it's coming up and approaching that cell. It's a big flat dendrite of one myelinated afferent fiber, and it makes close contact with an unusually large epithelial cell in the skin. These Merkel discs are sensitive to light touch and pressure. And on the bottom right, we have these Meissner's tactile corpuscles. We find these in the superficial dermis. They detect light touch, pressure, and low frequency vibration. They're phasic and they rapidly adapt. So we find these Meissner's tactile corpuscles. They're fairly large. They're found in eyelids and lips, fingertips, nipples, external genitalia. They're highly coiled and interwoven. So they're in really sensitive, hairless areas. So all of those areas that I just listed are super sensitive. If you take a look at your hand, there's a high concentration on the palm of your hand. So kind of rub the palm of your hand lightly with one finger and then flip your hand over and rub the back of your hand. And you'll notice that the back of your hand is less sensitive than the palm. For obvious reasons, right? We need our palm to be able to respond to read Braille and pick up things for pressure, that type of thing. We also have subcutaneous fine. So we also have subcutaneous fine touch. So these have larger receptor fields. This information is carried on the A beta fibers. So a couple examples of these are the Pacinian lamellated corpuscles on the left. These are in the deep dermis. They detect deep pressure and high frequency vibration. They're phasic, so they're fast adapting. And they are in kind of deep layers of collagen. So they're under such a thick layer that they adapt quickly because the distortion of the capsule is quickly relieved, right? There's a single dendrite that travels through the center, and this one dendrite is in layers of these collagen fibers. We see this in mammillary glands, genitalia, deep fascia, and joint capsules. We also have the Ruffini corpuscles on the right. They are deep in the dermis. They're sensitive to pressure, stretch of skin. These are tonic receptors with little adaptation. They discharge in response to static joint angles. The Ruffini capsule kind of surrounds, the capsule surrounds collagen fibers, and they're continuous with those of the surrounding dermis. The dendrites are inside the capsule, and there's when there's like tugging or twisting of the dermis, it twists this capsule and it compresses the dendrites.